The overall goal of this procedure is to deliver nucleic acids into the embryos of Ceratitis capitata, an important agricultural pest, to ultimately expand the toolbox for its control.
This method helps functionally characterize genes in a non-model organism, as well as generate transgenic strains.
The main advantage of this technique is that very low amount of nucleic acids are injected to achieve the desired effects.
Maintain all the life stages of the medfly colony at 25 degrees Celsius with 65% humidity and a 12-12-hour light-dark photo period with a photophase starting at 8 a.m. Insert a sponge strip through a small opening in the cage's base to provide the flies with water by means of capillary action. To establish a 6-liter cage, load one with between 1,500 and 2,000 pupae. For food, load the cage with about 45 grams of a 1-to-10 mixture of yeast and sugar. To collect eggs, place a plate filled with water below the cage. Females will deposit eggs through the mesh, which will then collect in the water. Because virgin females can oviposit, ensure that the eggs are all fertilized by waiting until the flies are at least a week old before collecting eggs. To collect eggs, filter the water with a fine strainer. Then, using a pasteuropipette, transfer the eggs into a plastic box loaded with about 0.5 liters of standard larval food.